Hi, in this video we are going to learn about biocatalysis, the first subtopic, which is properties of enzyme and mechanism of enzyme action. The learning outcomes are for students to be able to state the properties of enzymes, state 6 classes of enzymes according to IUBMB classification, explain how enzyme lowering activation energy, illustrate to explain the mechanism of enzyme action based on induced-FIT model, and then state the factors that affect the enzymatic reaction. First, what is enzyme? Enzyme is a molecule, a biological catalyst that is produced by living cells. So it must come from a living cell. So it is a biological catalyst that speeds up the rate of chemical reaction by lowering the activation energy. So whenever the enzymes are present in the system, the rate of chemical reaction will be sped up. So how does it speed up the chemical reaction? By lowering activation energy. Basically, in all enzymes, there will be two types of sites. The active site and the allosteric site. Active site is very specific. It is a specific site on the enzyme that allows a specific substrate to bind to it. So active site is for the substrate to bind, while allosteric site is another site other than the active site. Okay, other than the active site on the enzyme. So another substance will be bound to the allosteric site. The basic of enzyme reaction is for the enzyme, a substrate will bind to the active site of the enzyme to form enzyme substrate complex. And then after a chemical reaction occurs and the enzyme and the substrate will be changed into product. Okay, the substrate will change, but the enzyme will remain unchanged. We have to memorize the properties of enzyme. First, enzymes are globular protein. And then it acts as a biological catalyst that speeds up the rate of reaction by lowering the activation energy. Only a small quantity of enzyme are required to catalyze a reaction. Enzymes do not change after the reaction, meaning that enzyme is reusable. Enzyme is a reversible catalyst, so it catalyzes a reversible reaction. The active site of enzyme is specific to its substrate. Enzyme activity is maximum at optimum temperature and pH. So we know that these two parameters or two factors that will affect the enzyme activity. So an optimum temperature and pH means enzyme can be denatured or destroyed at extremely high temperature and extreme pH. Let's look at the specificity of the enzyme. The specificity, why it is become specific? Only certain substrate can bind to the active site because the shape of the active site complementary or only suitable to a certain substrate. Okay, because the complementary shape of the active site towards the substrate. So only a substrate that can fit to the active site's shape will be able to bind to the active site. When an enzyme is denatured by heat or a change in pH, the active site will change its shape. So the substrate cannot fit into the active site anymore. Okay. The classification of enzymes are recommended by International Union of Biochemistry and Molecular Biology, IUBMB. It is based on the type of reaction. The classification of enzymes are based on the type of reaction that the enzyme catalyzes. So the naming of the enzyme, basically it uses the name of the substrate and then it add up ASE suffix. Dia punya penghujung ada ASE. For example, sucrase meaning the substrate is the sucrose and then ASE. So sucrase. Protease means protein, enzyme that act on protein. Lipase is the enzyme that act on lipid. There are 6 classes of enzymes. We have lyase, transferase, ligase, oxidoreductase, hydrolase, and isomerase. Let's look at oxidoreductase. Oxidoreductase enzyme catalyzes the transfer of oxygen and hydrogen atom. Oxidoreductase comes from the word oxidation and reduction. So, in oxidation and also reduction, it involves the transfer, the acceptance or the removal of oxygen and also hydrogen atoms between substrates. So, oxidoreductase catalyzes the transfer of oxygen and hydrogen atoms between substrates. So it involves oxidation and reduction reaction. So these enzymes usually need coenzymes such as FAD, NAD+, and NADP+. The example of this enzyme is dehydrogenase, meaning that it removes hydrogen. Or this one, oxidase, it catalyzes the oxidation process. Malate dehydrogenase is a good example of an enzyme. The substrate is the malate. So what does it do? It dehydrogenates. It removes hydrogen from malate. Hydrogen from malate is removed and transferred to NAD+. So NAD+ is reduced to NADH plus H+. Remember that when a substance, a molecule receives hydrogen, it is said to be reduced, while malate will be oxidized because it lose hydrogen atoms and then oxidized to oxaloacetate. Succinic dehydrogenase, the substrate is the succinic acid or succinate. The enzyme transfer the hydrogen, catalyze the transfer of hydrogen from succinic acid to the FAD. So FAD is reduced to FADH2, while succinic acid will be oxidized to fumaric acid. Transferase enzyme. The word transfer, so this enzyme catalyzes the transfers of a functional group such as methyl, amino, and also phosphate group from one substrate to another substrate. The example is phosphorylase, transaminase. Hexokinase enzyme catalyzes the transfer of phosphate group from ATP received by glucose. So glucose is phosphorylated to glucose 6-phosphate, while ATP, since it's loose phosphate, it is converted from ATP to ADP. Hydrolase. Hydrolase enzyme catalyzes the breakdown of chemical bond by adding water molecule. Okay, hydro. So by adding water molecule. So in this example, sucrase is the enzyme that catalyzes the addition of water to sucrose, thus convert the molecule into fructose and alpha glucose. So the H2O is hydrolyzed, okay, hydrolyzed, and then the molecules will be received by fructose and alpha glucose, because sucrose is a disaccharide. So disaccharide will be split into fructose and alpha glucose. Ligase. Ligase enzyme catalyzes the addition or removal of group of atoms to a substrate. Decarboxylase, removal of carboxyl. Pyruvate decarboxylase. So in this equation, in this process, the pyruvate will lose a carbon molecule in the form of carbon dioxide, and then it will be converted to ethanol. Another example is RUBP carboxylase. In this case, in this process, RUBP, ribulose bisphosphate, will receive carbon dioxide molecule, which means the enzyme catalyzes the addition of carbon dioxide to RUBP and form 2-glycerol phosphate. Isomerase enzyme catalyze the rearrangement of atoms in a substrate molecule, which is the conversion of an isomer. Because the molecular formula does not change, but the position of the phosphate is changed from glucose 1-phosphate. Phosphate is located at the first carbon, and then it is being rearranged. The phosphate is transferred to the 6th carbon in the same molecule. So the enzyme catalyzes the rearrangement of the phosphate group from glucose 1-phosphate to glucose 6-phosphate. Ligase catalyze the formation of new bonds between two substrate molecules by using energy from ATP. So ligase form new bonds. For example, amino acid and tRNA. Okay, tRNA in the process of translation. So the enzyme is amino acid tRNA synthesis. ATP is involved because it provides energy to form the new bond. Okay. Let's look at activation energy. The symbol is EA. Energy, activation. Activation energy is the minimum amount of energy required by a reactant or a substrate to start a chemical reaction. So how does enzyme lowers the activation energy? When substrate bind with the enzyme at the active site, it will bring substrate closer to each other, towards the active site, or substrate and the substrate. So enzyme facilitate either the breaking or the formation of bond in the substrate to form product. For example, if the hydrolase will break the sucrase into a fructose and glucose. So it is the enzyme facilitate breaking of bond in the substrate. So more powerful collision between the reactants, the breakdown of bond will occur faster. So energy will be released and product will be formed. Mechanism of enzyme action. This is based on the specific conformation of the active site. Conformation also have similar meanings with the shape. Enzyme have specific shape with the active site, where a specific substrate will bind at the specific active site of the enzyme to form enzyme substrate complex. So substrates are brought closer to each other. For example, if a glucose should bind with another glucose to form maltose. Then enzyme facilitate the breaking of old bond between substrate and formation of new bonds between the product. So activation energy is lowered and product is formed. This graph shows an enzyme rate reaction, a hyperbolic curve. Right, at A point here, there is only a few substrate, so the rate of reaction is low. And then B, it can have more substrate here. This is low substrate concentration. This has higher substrate concentration. So more substrate means higher rate of reaction. But at point C, even though we have more substrate concentration, but there will be no increase in the rate of reaction because there is no more free active site. The enzyme, all of the active site of the enzymes are occupied by the substrate. When we have to discuss about enzyme action at this level, we are going to discuss based on induced fit model. Induced fit meaning something's happen, then the shape of the active site becomes fitter ataupun will be more compatible with the substrate. Okay, because the attachment of the substrate will induce the conformational change, the change of shape of the active site of the enzyme until the active site fits the substrate. Let's look at the diagram. The enzyme here, the active site of the enzyme does not have a specific shape. The active site is specific, but the shape is not exactly compatible with the substrate shape. So, but then because the substrate is specific to the active site, a bond will form between the substrate and the active site. So once the substrate is attached to the active site, then the enzyme active site will be changed to follow the shape of the substrate. Okay, so that is what we call as when attachment of substrate induce the change of the shape of the active site of the enzyme until the active site fits with the substrate. So memorize this. The active site of an enzyme is not fully complementary. Tak fit sangat ataupun tidak melengkapi, tak betul-betul melengkapi bentuk substrate. So when the substrate collides with this active site of the enzyme, or enzyme collides with the substrate molecule, so the substrate will bind to the active site. Okay, the substrate will bind to the active site. The binding of the substrate towards the active site will induce mengaruh sedikit perubahan pada bentuk tapak aktif enzyme. So the binding induce a slight change in the conformation of active site of enzyme. Conformation is shape. So the active site change the conformation and becomes barulah fully complementary with the substrate. So enzyme substrate complex is formed enable the enzyme to carry out their catalytic function, thus produce product, and then active site of enzyme will change back into its original conformation. So let's look at factors that affect enzymatic reaction. We learned about temperature, pH, substrate concentration, and also the presence of cofactor. The effect of temperature. At low temperature, the rate of reaction occurs slowly. Because why? At low temperature, the movement of the molecules is slow. So the collision, pertembungan antara substrate dan juga enzyme, the collision between the substrate and the enzyme active sites is low. So an enzyme also is considered as inactive at a very low temperature. As temperature increase, the rate of reaction will increase, because with the increase of temperature, the molecules vibrates more. The movement of molecules increase, so the vibration will cause or the movement of the molecule will cause collision between substrate and active sites to increase. Kan dia akan melangkah. Until an optimum temperature is achieved, which the rate of reaction is at maximum. Alright. Beyond optimum temperature, the active site, the enzyme will become denatured lah. Jadi beyond optimum temperature ni maksudnya suhu nak makin naik lagi. Contoh for example, human enzymes mostly works optimally at 35 degrees Celsius to 37.5 degrees Celsius or 40 degrees Celsius. Beyond, if we achieve or the temperature rise to more than 40, then something will be damaged in our body. Some of the enzyme will be damaged of the high temperature. That's why when we have a high fever, it is advisable for us to try to reduce our body temperature. While the thermophilic bacteria, the enzyme for thermophilic bacteria, a bacteria that live in a hot spring, is 70 degrees Celsius. So different species will have different optimum temperature. The higher temperature, beyond optimum temperature, reaction rate of reaction will decrease. At 60 degrees Celsius, usually the reaction will stop, and then the enzyme is denatured. Okay, what makes the protein denatured, the enzyme denatured? What makes the enzyme denatured? Because enzyme is a protein. The change in the conformation of active sites is because of the breaking of ionic bond, the hydrogen bond, the disulfide bridge, the hydrophobic and van der Waals interaction. Remember that we learned protein has, there are 4 levels of protein. The primary, secondary, tertiary and quaternary. The tertiary protein and quaternary protein is a globular protein, and they have a 3D shape because of these bonds: ionic bond, hydrogen, disulfide bridge, hydrophobic and van der Waals interactions. So with a higher temperature, it will cause the vibration to increase, thus will break the bonds that makes up the 3D structure, the globular shape of the enzyme. So when these bonds are broken, the tertiary and the quaternary levels of the protein will be opened up. Okay, dia akan terbuka and then they will become primary structure. So substrate can no longer fit into the active site of the enzyme. So this is when we say that the enzyme is denatured. Okay. Effects of pH. Different enzyme function efficiently within a different pH range, and the range is very narrow. Right. For example, pepsin. They works well optimally at pH 2, while salivary amylase. Amylase is the enzyme that breaks down the amylose, the starch. Usually can be found in our saliva, dalam mulut kita. So the amylase works well around pH 7.2. Different with pepsin, which meets an acidic environment. So optimum pH is a pH at which rate of reaction is at maximum. Itu optimum. Bila optimum pH, rate of reaction is maximum. Bila rate of reaction is maximum, means that it is the optimum pH. Above or below optimum pH or beyond the pH range, the rate of reaction will decrease. Okay, deviation from optimum pH range. Okay, dia deviation maksudnya kalau optimum ni, di sini optimum, jadi duduk di sini. Sorry ini maksimum So ini adalah optimum. So kat sini rate of reaction low. Sini low. Deviation from optimum pH range results in excess hydrogen ions or hydroxyl hydroxyl ions. So excess hydrogen means it is acidic. Excessive hydroxyl ion means it is basic. So that alters the acidic or basic or the functional group or the side chain of the amino acids in the enzyme. So remember amino acid Okay, this is the carboxyl group, we have the amino group, we have hydrogen, and we also have the side chain of amino acid. So excessive hydrogen ions will affect first it will be received by the amino terminals, and then it will affect the hydrogen, it will affect the side chain. Okay. Now, while hydroxyl acid has hydroxyl ion, excessive of OH minus, it makes the amino acid to remove its hydrogen from the carboxyl group or affect the side chain. Okay. So, causing ionic or hydrogen bonds to be broken, thus change the conformation of the active site. So, enzyme is denatured, substrate cannot bind to active site, and lower the rate of reaction. If pH is too low or too extreme, enzyme will denature. Too basic, enzyme will denature. Okay, different enzyme will have different optimum pH. Substrate concentration. At low substrate concentration, rate of enzyme reaction will increase. S increase with increasing substrate concentration. Okay. As we add up the substrate concentration. Because at low substrate concentration, not all active sites bind with the substrate. So some of the active site will be available to bind with another substrate. As the substrate concentration increase, the rate of reaction increase. So collision between substrate and active site also increase. More active site binds with the substrate. until if we continue to add up the substrate up to a point, there will be no more active site because all of the active sites are bound to the substrate. So at this point, which is called as point of saturation, the rate of reaction becomes constant. So any further increase of the substrate will no longer affect the rate of reaction because all of the active sites are bound to the substrate. Okay, cofactors. Cofactor is a non-protein substance. So usually cofactor present within the active sites. Sometimes they are also needed for proper functioning of the enzyme. Without it, the enzyme will not function properly. In a way, it is needed to activate certain enzyme. Without the cofactor, the enzyme will not be activated. Thus, the presence of cofactor will increase the rate of reaction. Cofactors can be permanently bound to the active site or loosely bound to the substrate during the catalysis. Why are the products separated from the enzyme at the end of reaction? Because the product is no longer fit to the enzyme's active site or does not complement with the active site because the active site is specific to the substrate, not the product. So the product will be released. Thank you.